Students, today we will learn about biomolecules. Let's begin with biomolecules. The living matter is composed of mainly six elements: carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. These elements together constitute about 90% of the dry weight of the human body. Several other functionally important elements are also found in the cells. These include calcium, potassium, sodium, chlorine, magnesium, iron, copper, cobalt, zinc, molybdenum, and selenium. And now let's know about carbon, a unique element of life. Carbon is the most predominant and versatile element of the life. It possesses a unique property to form infinite number of compounds. This is attributed to the ability of carbon to form stable covalent bonds and carbon-carbon chains of unlimited length. It is estimated that about 90% of compounds found in living system invariably contain carbon. Now let's know about chemical molecules of life. Life is composed of lifeless chemical molecules. A single cell of the bacterium like Escherichia coli, or in short E. coli, contains about 6,000 different organic compounds. It is believed that man may contain about 1 lakh different types of molecules, although only a few of them have been characterized. Now let's know about complex biomolecules. The organic compounds such as amino acids, nucleotides, and monosaccharides serve as a monomeric units or building blocks of complex biomolecules like proteins, nucleic acids means DNA and RNA, and polysaccharides respectively. The important biomolecules, macromolecules with their respective building blocks and major functions, we will discuss now. Suppose protein, its building block is amino acids. Its major function is fundamental basis of structure and function of cells. And next is DNA, deoxyribonucleic acid. Its a building block is deoxyribonucleic acids, uh, deoxyribonucleotides, and its major function is repository of hereditary information. Next is RNA, ribonucleic acid. Its building block is ribonucleotides, and its major function is essentially required for protein biosynthesis. Next point is polysaccharide. Next biomolecules is polysaccharide, that is glycogen, monosaccharides. Its building block is monosaccharide, that means glucose. Its function is to storage form of energy to meet short-term demands. Now biomolecules, lipids. Building block is fatty acids and glycerol, and major function is storage form of energy to meet long-term demands, structural components of membrane. Now let's know about structural hierarchy of an organism. The macromolecules like proteins, lipids, nucleic acids, and polysaccharides form sapro, uh, sup, supramolecular assemblies, example membranes, which in turn organize into organelles, cells, tissues, organs, and finally the whole organism. Now let's know about chemical composition of man. The chemical composition of a normal man weight 65 kg. Water is the solvent of life and contributes to more than 60% of the weight. This is followed by protein, mostly in the muscle, and lipid, mostly in the adipose tissues. The carbohydrate content is rather low, which is in the form of glycogen. The basic information on the various biomolecule is essential for a better understanding of the concept of biotechnology. The chemistry of carbohydrates, lipids, and proteins is we will now discuss the biomolecules, namely nucleic acid, DNA, and RNA, which are directly relevant to biotechnology. And now let's begin with carbohydrates. Carbohydrates are the most abundant organic molecules in nature. They are primarily composed of the elements carbon, hydrogen, and oxygen. The name carbohydrate literally means hydrates of carbon. Carbohydrates may be defined as polyhydroxyaldehydes or ketones or compounds which produce them on hydrolysis. The term sugar is applied to carbohydrates soluble in water and sweet to taste. Let's know about the function of carbohydrates. Carbohydrates participate in a wide range of functions. They are most abundant dietary source of energy for all organisms. Carbohydrates are precursors for many organic compounds like fats, amino acids, etc. Carbohydrates as glycoproteins and glycolipids participate in the structural and cell membrane and cellular functions such as cell growth, adhesion, and fertilization. Next point is carbohydrates also serve as the storage form of energy to meet the immediate energy demands of the body. Now let's know about the classification of carbohydrates. Carbohydrates are often referred to as saccharides. Example, saccharon means sugar, and they are broadly classified into three groups. First is monosaccharide, oligosaccharide, and polysaccharides. This categorization is based on the number of sugar units. Mono and oligosaccharides are sweet to taste, crystalline in character, and soluble in water. Hence, they are commonly known as sugars. Now, let's know about monosaccharides. Mono means one, and are the simplest group of carbohydrates, are often formula CnH2O whole N, and they cannot be further hydrolyzed. Based on the number of carbon atoms, the monosaccharides are regarded as triages, means 3 carbon, tetrages, that is 4 carbon, pentages, that is 5 carbon, hexages, that is 6 carbon, and heptages, that means 7 carbon. These terms along with functional group are used while naming monosaccharides. For instance, glucose is a aldohexose while fructose is a ketohexose. Now let's know about oligosaccharides. Oligo means few, contains 2 to 10 monosaccharides molecule which are liberated on hydrolysis. Based on the number of monosaccharide units present, the oligosaccharides are further subdivided into disaccharides and trisaccharides, etc. Now let's know about polysaccharides. Poly means many, are polymers of monosaccharide units with higher molecular weight. They are usually tasteless, that means non-sugar, and form colloids with water. Polysaccharides are of two types, that is homopolysaccharides and heteropolysaccharides. Now let's know about monosaccharides. Stereoisomerism is an important character of monosaccharides. Stereoisomers are the compounds that have the same structural formula but differ in their spatial configuration. A carbon is said to be asymmetric when it is attached to 4 different atoms or groups. The number of asymmetric carbon atoms determines the possible isomers of a given compound, which is equal to 2 to the power n. Glucose contains 4 asymmetric carbon and thus has 16 isomers. Now let's know about glyceraldehyde, the reference carbohydrate. Glyceraldehyde is the simplest monosaccharide with the one asymmetric carbon atom. It exists as two stereoisomers and has been chosen as a reference carbohydrate to represent the structural Structure of all other carbohydrates. Now D and L isomers. The D and L isomers are minor images of each other. The spatial orientation of hydrogen and OH, hydroxyl group and OH groups on the carbon atom, that is carbon C5 for glucose, that is adjacent to the terminal primary alcohol carbon determines whether the sugar is D or L isomer. If the OH group is on the right side, the sugar is of D series, and if the on the left side, it belongs to L series. The structures and D and L glucose based on the reference monosaccharide D and L glyceraldehyde are under consideration. It may be noted that the naturally occurring monosaccharides in the mammalian tissues are mostly D configuration. The enzyme machinery of the cells is specific to metabolize D series of monosaccharides. Now let's know about optical activity of sugars. Optical activity is a characteristic feature of compounds with asymmetric carbon atom. When a beam of polarized light is passed through a solution of the of an optical isomer, it will be rotated either or to the right or left. The term dextrorotatory and levorotatory are used to compounds that respectively rotate the plane of polarized light to the right or to the left. Now let's know about glycosides. Glycosides are formed when the hemiacetal or hemiketal hydroxyl group of a carbohydrate reacts with a hydroxyl group of another carbohydrate or a non-carbohydrate, example methyl alcohol, phenol, glycerol, etc. The bond so formed is known as glycosidic bond, and the non-carbohydrate moiety is referred to as aglycon. Now let's know about derivatives of monosaccharides. Derivatives of monosaccharides. There are several derivatives of monosaccharides, some of which are physiologically important. First is amino acids. When one or more hydroxyl groups of the monosaccharides are replaced by amino groups, the products formed are amino sugars. Example D-glucosamine, D-galactosamine, and they are present as constituents of heteropolysaccharides. Now let's know about deoxysugars. There are These are the sugars that contain one oxygen less than that present in the parent molecule. The groups CHOH group CHOH and CH2OH become CH2 and CH3 due to the absence of oxygen. D2 deoxyribose is the most important deoxysugar since it is a structural constituent of DNA. L next is third point is L ascorbic acid, that is vitamin C. This is a water soluble vitamin, the structure of which closely resembles that of a monosaccharide. Now let's know about disaccharides. Among the oligosaccharides, disaccharides are the most common, and as is evident from the name, a disaccharide consists of two monosaccharide units held together by a glycosidic bond. They are crystalline, water soluble, and sweet to taste. The disaccharides are of two types. One is reducing and another is non-reducing. Reducing disaccharides with free aldehyde or keto group. Example maltose, lactose, etc. Second is non-reducing. Non-reducing disaccharides with no free aldehyde or keto group. Example sucrose, tetra, trehalose. Now let's know about polysaccharides. Polysaccharides consist of repeat units of monosaccharides or their derivatives held together by glycosidic bonds. They are primarily concerned with two important functions, structural and storage of energy. Polysaccharides are of two types, homopolysaccharides, which on hydrolysis yield only a single type of monosaccharide. They are named based on the nature of the monosaccharide unit. Thus, glucans are polymers of glucose, whereas fructosands are polymers of fructose. Second is heteropolysaccharides on hydrolysis yield a mixture of a few monosaccharides or their derivatives. Now let's know about homopolysaccharide, that is starting with starch. Starch is the carbohydrate reserve of plants which is most important dietary source for higher animals including man. High content of starch is found in cereals, roots, tubers, vegetables, etc. Starch is a homopolymer composed of D-glucose units held by alpha glucose glycosidic bonds. It is known as glucosan or glycan. Glucan. Starch consists of two polysaccharide components, water-soluble amylose and a water-insoluble amylopectin, that is 80 to 85%. Chemically, amylose is a long unbranched chain with 200 to 1000 D-glucose unit held by alpha glycosidic linkages. Amylopectin, on the other hand, is a branched chain with alpha glycosidic bonds at the branching points and alpha, that is 1 to 4 linkages everywhere else. Amylopectin molecule containing a few thousand glucose units looks like a branch tree. Now let's know about glycogen. Glycogen is the carbohydrate reserve in animals, hence often referred to as animal starch. It is present in high concentration in liver followed by muscle, brain, etc. Glycogen is also found in plants that do not possess chlorophyll, example yeast, fungi. The structure of glycogen is similar to that of amylopectin with more number of branches. Glucose is the repeating unit of glycogen joined together by alpha 1 4 glycosidic bond and alpha glycosidic bonds at branching point. Now let's know about cellulose. Cellulose occurs extensively in plants and it is the most abundant organic substances in plant kingdom. It is a predominant constituent of plant cell wall. Cellulose is totally absent in animal body. Cellulose is composed of beta D glucose units linked by beta glycosidic bonds. Cellulose cannot be digested by mammals, including man, due to lack of enzyme that cleaves beta glycosidic bonds, that is alpha amylase breaks alpha bonds only. Certain ruminants and herbivorous animals contain microorganisms in the gut which produce enzymes that can cleave beta glycosidic bonds. Hydrolysis of cellulose yield a disaccharide cellobiose followed by beta D glucose. Cellulose, though not digested, has great importance in human nutrition. It is a major constituent of fiber and in non-digestible carbohydrate. The functions of dietary fiber include decreasing the absorption of glucose and cholesterol from the intestine, besides increasing the bulk of feces. Now let's know about heteropolysaccharides. When the polysaccharides are composed of different types of sugars of their derivatives, they are referred to as heteropolysaccharides or heteroglycans. Now let's know about mucopolysaccharides. These are heteroglycans made up of repeating units of sugar derivatives, namely amino acids and uronic acids. Mucopolysaccharides are more commonly known as glycosamine glycans. GAG, acetylated amino groups besides sulfate and carboxyl groups are generally present in GAG structure. Some of the mucopolysaccharides are found in combination with proteins to form mucoproteins or mucoids or proteoglycans. Mucoproteins may contain up to 95% carbohydrate and 5% protein. Mucopolysaccharides are essential components of tissue structure. The extracellular spaces of tissue, particularly connective tissue, cartilage, skin, blood vessels, tendons, consist of collagen and elastin fibers embedded in a matrix or ground substances. The ground substance is predominantly composed of GAG. The important mucopolysaccharides include hyaluronic acid, chondroitin, 4-sulfate, heparin, and darmatan sulfate and keratin sulfate. Now let's know about lipids. Lipids are of great importance to the body as cheap concentrated storage form of energy. Besides their role in cellular structure and various other biochemical functions.